push the recording button here. Just before we start today, I want to take a moment and publicly thank Margaret Graham at the University of Edinburgh, who helped me to both organize this series and who acted as the host for several of the talks. So thank you, Margaret, for all of your efforts in this. And thank those of you who have stuck with us for the whole 11 talks. I hope you've enjoyed this. I also want to mention that we're going to be having another lecture series starting probably late September, so you can watch our website, and that will be on environmental problems or environmental issues associated with small island nations and coastal marine regions. So I think that should be an exciting series. That's going to be organized by Professor Chris Metcalf at Trent University. This brings us to the end of our series on nanoparticles. This is our 11th talk, and it's, I think, a good way to end our talk. I'm very pleased to introduce our speaker today, Professor Sijin Liu, who received his PhD degree from the Institute of Developmental Biology and Genetics of the Chinese Academy of Sciences in 2004. He continued his postdoctoral training in hematology and oncology at MIT and Children's Hospital Boston, the Harvard Medical School, from 2004 to 2008. And after that, he became an instructor at Tufts University School of Medicine, starting in 2008. He's currently a professor at the State Key Laboratory of Environmental Chemistry and Ecotoxicology, which is part of the Research Center for Eco-Environmental Sciences of the Chinese Academy of Sciences. His research interests include environmental toxicology and nanotoxicology, and translational research into nanomaterials in biomedicine. So it's a great pleasure to have you with us today, Professor Liu.
Thank you. So thanks, Doug, for the kind introduction. And first, I'd like to thank the organizer, including Margaret, Doug, for inviting me to give this talk. Today, I'm going to talk on the passage of exogenous fine particles in the body at the organ and cellular levels. So today, I'm going to talk two parts. Here's the layout of the talk. The first part would be the passage of PM, namely fine particles in the body of humans and animals. And the second part would be the transport of pollutants by these PM particles. A introduction about this background. Pollution is still a huge burden on health and mortality globally, as indicated by a most recent estimation on the risk or causes for death worldwide, published recently in Lancet. And of these risk factors, air pollution certainly is a leading contributor to the mortality. This is a global statistics about the mortality contributed by different risk factors. We can see similar trends are also observed in China. We can visualize the different contributors to the mortality similar to the global data. Still facing the high levels of PM pollution. So as we can see in the left panel, the PM pollution like PM 2.5 is still a great concern in China. So we can see here the concentration of a PM concentration is PM pollution is still very high here. So ranging from 60 to 80 in China, which is still much higher than WHO instructions. And we also observed high risk of health effect ranging from 60 to 80. So which should suggest a great contribution of air pollution to the detrimental health effects. So today, considerable progresses have been obtained in the past years, leading to improved understanding on PM-associated health effects, such as neurotoxicity, lung cancer, COPD, and heart disorders. Huge challenges in this field. For example, the biodistribution and the target size in the body, in particular, whether particles could cross the biological barriers, like different biological barriers in the brain, in the joint, or also like in the embryo, whether this particle could cross these biological barriers to reach different organs. To address these challenges, we finished some experiments to specifically look at the brain and the joint. So firstly, we examined the particles in the human cerebrospinal specimens. So we collected this CSF specimens from hospitalized patients. And then we used different high-tech methods, including HIDF, STEM, microscopy to look at the exogenous particles in these specimens. Surprisingly, we found 8 out of 25 specimens. Namely, we found exogenous particles in 8 patients. So in these biological specimens. So in more details, we uncovered some exogenous particles that have been identified in previous reports, such as calcium-containing particles, such as calcite and aragonite. These particles have been reported in previous studies, and also including like iron-containing particles, which are also reported in previous studies. Similarly, silicon-containing particles such as silicon dioxide, which was also identified in like blood or other biological specimens. So we also identified silicon-containing particles in human CSF specimens. Interestingly, we also uncovered some new exogenous particles in human CSF that have not reported previously in human specimens, including Maillard particles. So Maillard particles is particles which used in like ceramic pans. So this kind of ceramic pans are widely used like in different handicrafts, including like traditional Chinese folk handicrafts. So it's widely used in China. So it's like a pen used for different like painting. So in this case, we would assume this case would be like occupational exposure, as this patient is a professional worker on traditional Chinese folk handicrafts. Additionally, another type of exogenous particles are also identified in human CSF specimens, which is titanium dioxide. This type of titanium dioxide anesthase is the first time identified in human specimens, which has not been previously reported yet. Now, titanium dioxide is widely used in different food packages, food additives, and other types of like daily used products. So in this case, we would assume that it would be like occupational or environmental exposure to have this internal exposure of titanium dioxide. Consistently, we also identified the same types of exogenous particles in human blood samples, including Maillard samples and also like silicon dioxide samples in the same patients. So this data would suggests that the particles may cross the bloodstream from the brain-blood barrier, namely BBB, for final localization in the ventricles of the brain. So to test this hypothesis, we did a lot of in vivo and in vitro experiments to test our hypothesis. So first, we found a number of animal experiments to substantiate the findings in humans. So we use animal models. So we look at the translocation of particles in blood from inhalation, and also we specifically look at the translocation of particles in the brain. So we could obviously see the translocation of these particles from the lung into circulation through damaging the blood-air barrier. And we also identified the particles, the same particles in the blood samples. And we afterwards look at the localization of these particles in the brain. So eventually we visualize these exogenous particles in the brain tissue. So we would conclude particle transport into the brain and also damage the BBB. To confirm these findings, we employed a number of different particles in these experiments. So we obtained similar results by in situ characterization of these particles through TEM, and we also look at the permeability of the brain by quantifying the Evans blue dye. We could see here the increased permeability of the brain tissue after exposure to these particles. Moreover, we established the in vitro BBB model, and we also look at the translocation of the particles through the in vitro BBB model. So we then characterize these particles through TEM and quantify the particles through ICP mass. So we could obviously see these particles translocated, and also we visualized the particles in particle form and also in ion form. So we obtained this by different types of particles like metal particles, and which are widely used in a number of applications. More importantly, in this study, we used the isotope, namely iodine 131 radioisotope, to label the carbon black fine particles to study the particle kinetics of these particles in the brain and other organs. On one hand, these results substantiated the biodistribution of the fine particles in different organs, similar to the results obtained above. And on the other hand, a much longer half-life were observed in the brain, like the half-life in the brain is 2.8 days, in contrast to 0.8 days in the liver, suggesting that the longer retention and the difficulty in clearance of these fine particles from the brain, which also suggested the long-term toxicity to the brain. The first part of the story, we used the comprehensive in vitro and in vivo animal and human experiments demonstrated the passage of inhaled exogenous particles into the brain. So we set up different levels experiments to confirm translocation of the particle from inhalation into the lung, into the circulation, and finally into the brain. So which was published just 2 or 3 days in PNAS. And then we used the same strategy to look at the particles in human joint effusion specimens from hospitalized patients receiving joint surgeries. So very similarly, we also identified some exogenous particles in human joint effusions in these patients. So in more detail, we recognize exogenous particles in human joint effusions, like commonly found exogenous particles in the human specimens, like in the lung, fluid, in the blood, like silicon-containing particles, and also iron-containing particles in these joint effusion specimens. We also set up the animal experiments to validate the particle translocation from the lung into the blood circulation and finally localized in the joint. So we observed the step-by-step translocalization of these particles in the lung, blood, and finally in the joint. We look at the particles through TEM and also through H&E staining to look at the histological alternations, like the inflammation to the synovium, and the collagen is also observed with inflammation, and we identify the particles within the tissues. And more importantly, like we did for the brain, so we use the iodine 131 isotope labeled particles to track the particles translocation, and the results suggested iodine 131 labeled particles was detected in the joint, and also a longer half-life in the joint, suggesting longer and lasting effect to the joint. So for the second part of the story, so we identified the passage of the inhaled exogenous particles from the lung into the joint. And this is like a diagram for the story. So this story was just recently published in Nano today. So, this is the first story. And the second story would be like the transport of pollutants by this PM. So we look at the translocation of these fine particles to carry the pollutant, and also we look at a different toxicity profile of this pollutant in contrast to free organic pollutants. All physicochemical properties together with associated pollutants collectively dictate the toxicity features of the fine air particles, as shown in this schematic. In the current stage, the toxicity features underlying the exposure of pollutant-laden carbon particles are not well understood. In other words, what are the changed toxicity profiles for the absorbed organic pollutants in different, which may be different from free organic pollutants. So to address this challenge, we here on the toxicity of pollutant-laden particles in contrast to free pollutants to different types of cells in the lung, including the lung epithelial cells and the lung macrophages. We prepared parental biochar from particles, and particles absorbed with pH at different concentrations containing like 10 or 100 microgram per gram pH, namely alpha BP, respectively. We observed significant phagocytic differences between macrophages and epithelial cells responding to these fine particles. As macrophages, which could visualize here, macrophages revealed massive phagocytosis of these fine particles. By contrast, as shown here, lung alveolar epithelial cells did not show observable phagocytosis of these these fine particles independent of a pH loading, we could only see the particle outside of the cells. But in contrast to the fine particles, does this mean? This means specifically particles function as a vehicle to deliver these pollutants into the macrophages. And in other words, through the Trojan-Horse effect, these fine particles could carry these pollutants into the macrophages. And just like this imaging show, it's just like a Trojan-Horse effect for the carbon fine particles to carry and deliver organic pollutant into the cells, into the macrophages. Once inside the macrophages, so the pollutant could be released and incur toxicities to the macrophages. Different from macrophages, epithelial cells do not uptake the fine particles. There's no particles observed inside the epithelial cell, but only particles are observed outside of the epithelial cells. So in other words, it's like but this particle outside of the cells could continuously release pH, and release pH could further reach the epithelial cells. In other words, it's like a reservoir effect through which enhanced exposure of pH could occur to these epithelial cells. So to substantiate this observation, an HR receptor system was employed to study the response to pH signaling of dissolved alpha BP from these fine particles. So we could visualize here, this suggested continuous releasing of alpha BP from particles to activate intracellular HR signaling. So just like the reservoir effect in the lung fluid, so the organic pollutant absorbed on fine particles could be continuously released from these particles and in the lung fluid, and where the organic pollutant could afterwards reach the cells and induce the additional toxicities to the epithelial cells. Summarize, we unearthed the increased toxicity of alpha BP to the lung macrophages and the lung epithelial cells by the fine particles through different mechanisms. For the macrophages, it resides in Trojan-Horse effect, but for the epithelial cells, it serves like a long-term contaminant reservoir. So this work was published in EST letter in 2021. In the meanwhile, alpha BP absorption also changes the physicochemical properties of the particle per se, mainly reflected by the decreased per surface area and per volume. So we wonder what happened to these fine particles after absorption of the alpha BP. We want to address the change toxicities of these fine particles, which may be different from parental particles. But we did not observe the significant changes to the other physicochemical properties after absorption of this organic pollutants pH. But we observe significant toxicity changes to the fine particles after absorption with the pH. Alpha BP absorption, in fact, increases the cytotoxicity of fine particles, as evidenced by the reduced survivability, especially for the high concentration absorption of a pH, and also demonstrated by the PI staining, as we can visualize here. Alpha BP absorption, especially at a higher concentration, induce greater cell death compared to the parental fine particles. Interestingly, at the cellular level, alpha BP absorption increases the interaction between fine particles and the cell membrane, as manifested by the massive changes to the plasma membrane in the TM imaging, especially for the higher concentration alpha BP absorption onto the fine particles. We also use the FITC-BSA labeling to visualize this interaction, as we can see in the fluorescent imagings, especially at the higher concentration, we could visualize, massive endocytosis of these fine particles and also a number of particles associated on the plasma membrane, visualized by this green color. So compared to this parental like particle, we would observe the increased association of pH-laden fine particles towards cell membrane. And especially at the single cell level, we used AFM to show the dramatic changes of cell morphology of these macrophages. As we can see, quantified data here, nearly 50% of the reduction of cellular height for the treatment of alpha BP loading particles at higher concentration of alpha BP loading compared to the untreated cells and compared to the cell treated with parental particles. This active interplay between alpha BP laden fine particles and the cells caused membrane damages and RS production and CDS measurement also visualized the plasma membrane injuries, and also we quantified the RS production, also verified the injuries to the cells, like cells, especially for the cell membrane. In the end, alpha BP bearing particles cause the enhanced cell autophagy, leading to elevated cell death relative to parental fine particles. We could visualize the increased concentration of ILC3, 1 and 2, and also the ratio between RC3 and RC2 and 1. Additionally, these impairments also undermine the production of cytokines in macrophages, as evidenced by the compromised production of cytokines, especially for the pro-inflammatory cytokines like IL-6, upon exposure to the pH-laden fine particles compared to the untreated cells and also parental particle treated cells. To summarize, we here uncovered alpha BP bearing fine particles incurred greater cytotoxicity to macrophages than pristine fine particles through more active association of particles with the plasma membrane. So leading to enhanced interaction and endocytosis, and then afterwards increased cytotoxicities of leads pollutants to the cells. And this work was also recently published in IIES Nano. Here's the second story of the talk. Finally, I'd like to thank different funding sources from the Chinese government, and also thank the lab member for hard working, and also thanks the audience for your listening. Thank you very much.
Thank you very much, Professor Liu, for that fascinating and maybe I should say a little bit frightening talk. It was really very interesting. We can take some questions now if people have questions. You can either do it through the chat or you could by all means, open up your microphone and your camera if you've got a camera, and ask questions live. Maybe while people are thinking about questions, I could start and ask you one. Early on in your talk, you were talking about the half-life of the various particles in brain and other tissues. And you said that, yeah, so you're seeing half-lives of sort of anywhere from, say, 1 to 3 days.
Yeah.
What happens to those particles as they disappear? Are they changing their structure, becoming smaller? Are they dissolving? Are they just being eliminated in either urine or feces, or how are they being removed?
Yeah, so mostly these particles eventually will be cleared from the body through feces and other ways. These fine particles are very small, like a few nanometers. So we observe the aggregation of these particles in the body. So it's like it may not stay in single particle, but likely in aggregates, like aggregated form of by different, like a number of particles together. Because they are carbon particles, they may not be degraded easily. I would say mostly they will be secreted out of the body, like through either like a single particle or aggregated forms.
Okay, that makes sense. Thank you.
Yeah.
Do we have other questions? We have a question from Margaret Graham. Thank you for the excellent presentation. Margaret would like to ask if there might be other contaminants, such as organic or metal compounds associated with the biochars, and if so, how might their effect within cells be characterized?
Yeah, that's true. Yeah. So so we use the model particles to make the things simple. So in a simple way, otherwise it will be very complicated to look at different pollutants together, like organic metals together. So it will be very complicated to have a full understanding of the behavior of these pollutants inside of the cells. So we also performed like metal pollutants. We look at the association of this like calcium and also other metals to associated with fine particles. So compared to the organic pollutants, these heavy metals could be easily dissolved from this carbon fine particles. So we look at the dissolution. It could be easily like 1 or 2 days, so easily dissolved from particles, but we cannot conclude everything would be dissolved. There would be something still associated. There would be a dynamics between metals and the particles.
Yeah. I have another question for you.
Sure.
I could be wrong on this, but it seems to me that inflammatory diseases are much more common today than they might have been, say, 20 or 30 years ago.
Yeah.
To what extent do you think small particles of the kind that you've talked about getting into joints would account for the increases in inflammatory disease? So I did not have time today to specifically to talk about our huge population study. We look at the population with like more than 10 million people in Beijing to look at the visiting of doctors during the polluted days, PM polluted days, compared to the like clear sky days. So we observed like much more increased occurrence of like visiting seeing doctors in these polluted days of the joint disease, like the OA, so osteoarthritis in this, especially in these elder people.
This data have not been published yet, but we would assume there would be two ways, two mechanisms to explain this. One would be, like you said, the systematic inflammation would account for the occurrence of joint inflammation. That would be like systematic pro-inflammatory cytokines delivered to the joint. This would be the first mechanism. The second mechanism would be, as we discovered in this study, could be direct translocation of particles into the joint, where the particle could directly damage the, the synovial membrane, the collagen, what are other tissues. That would be the direct mechanism. So could be simultaneous two mechanisms existing together.
Okay, thank you. Do we have any other questions for our speaker? If not, I want to thank you once again, Professor Liu. You have provided a really excellent end cap to our series, sort of brought us full circle to where we can see the issues of nanoparticles, not only as we've seen in previous talks in environmental compartments, but directly impacting humans. Your work is really excellent, and obviously is going to have a lot of impact in terms of how we think about issues of nanoparticles in the environment. So I want to thank you once again. I want to thank those of you who've joined us today, and remind everybody that all of these talks are on our website, and you can go back and you can hear them at your leisure if you want to repeat any of them, or if you've missed any of the previous talks. And also to just once again remind you that we'll have another series in the fall in September. And so watch our website and hopefully you'll join us for that one as well. So thank you again, Professor Liu, and
Thank you.
Thanks to everybody, and thank you, Margaret.
Thanks, Doug. Thank you, Margaret. Bye-bye.
Goodbye, all.
Bye-bye.